Hello, everybody, and welcome. I am Kawsar Tallat. I'm an associate professor in the Department of International Health at the Johns Hopkins Bloomberg School of Public Health. And I am also an investigator on the Pfizer COVID-19 vaccine trial and a co-investigator on the AstraZeneca vaccine trial and a participant in the Moderna vaccine trial. So I've had lots of experience with COVID vaccines. And today I am going to talk about COVID-19 vaccines and how were they developed so quickly. So first of all, what I'd like to say is before the pandemic even started, there was a lot of groundwork that was laid in the preparation for the development of the next pandemic vaccine. There was a lot of work in early on in the structure-based vaccine design. And a lot of people have been working on rational vaccine development. There was basic science research into coronaviruses and other envelope virus spike protein structure. And more importantly, after understanding the spike protein structure, then there was work done at the Vaccine Research Center at the NIH to establish how to stabilize the spike protein so that it was in the best conformation for a vaccine. There was a lot of prototype vaccine development in advance of the next pandemic, and this was done at the NIH, but also spearheaded by CEPI and other organizations, and we'll talk about CEPI, and also a lot of research into the respiratory syncytial virus vaccines at the Vaccine Research Center was directly translatable to the development of the SARS-CoV-2 virus vaccine. So this has been work that has been going on for decades, but especially over the last 3 to 8 years prior to the pandemic, and really helped us hit the ground running when it came time to develop a pandemic vaccine. One of the organizations directly responsible for some of this preparation is CEPI, which is a coalition for epidemic preparedness and innovations. CEPI is a global alliance for financing and coordination. It was launched in 2017, and their mission is to accelerate the development of vaccines against emerging infectious diseases and to enable the equitable access to these vaccines during outbreaks. CEPI has been funding innovative vaccines and technologies, including the mRNA and adenovirus vector vaccines, by funding platforms that allow the switching of the viruses into established technologies. They funded, prior to the pandemic, the vaccine platforms that were later used to create the Moderna vaccine, the Janssen vaccine, which is Johnson & Johnson, the University of Oxford AstraZeneca vaccine, and the Novavax vaccine. So 4 of our lead vaccines for this pandemic virus. CEPI also funded mechanisms that speed up research and development and manufacturing, and most importantly, have a commitment to provide vaccines for the world. And they've been working really hard to try to live up to that commitment during the pandemic. Another factor in the early development of the vaccines was Operation Warp Speed, which was established by the Trump administration. And this was a US government initiative to accelerate the development, manufacturing, distribution of COVID-19's therapeutics and diagnostics or medical countermeasures. And they worked really hard, starting with about 14 candidates to align protocols for safety and efficacy. The goal was to get those 14 candidates down to somewhere between 3 and 5. And while they were doing the clinical testing, they were continuing the non-clinical testing of the vaccines and manufacturing, really importantly, manufacturing vaccines ahead of the results of the trials, so that if the trials proved to be successful, that the vaccine would be available for use right away, which is very unusual in the vaccine development world. So the Operation Warp Speed for the vaccines, what moved through the Coronavirus Prevention Network, or the CoVPN. They targeted at least 30,000 volunteers for each vaccine in terms of their phase 2-3 trials. They tried to harmonize the trials with one another so they could be comparable, although this was not completely done. But the plan was to do all of these trials as randomized placebo-controlled vaccine trials so that we could have data that is usable. The priority vaccine candidates in the COVID-PN network were the Moderna NIH vaccines, the Oxford AstraZeneca vaccine, the Janssen or Johnson & Johnson vaccine, Novavax vaccine, the Sanofi vaccine, and the Merck vaccine that was very quickly abandoned at some point. There were additional vaccines that were very critical in the development and the process that the government helped facilitate by buying doses, although they did not have a role in the clinical trial, and that was for the BioNTech Pfizer vaccine. This is just a timeline of how the mRNA 1273 vaccine this is just a timeline of how the mRNA 1273 vaccine was developed. And this is the vaccine that was created by the NIH and Moderna. And I'd like to point out Dr. Kizzmekia Corbett, who is a scientist who worked at the NIH, now at the Harvard School of Public Health, who did a tremendous amount of work on the SARS and MERS vaccines prior to the pandemic, helped determine the pre-fusion structure of the coronavirus spike protein, which was critical and instrumental in developing this vaccine. And as I mentioned earlier, CEPI had funded the vaccine development platform, the Moderna mRNA vaccine development platform for rapid vaccine development, as well as the adenovirus vector vaccine. So all of this groundwork had been laid for several years prior to the pandemic, so that on December 31st, 2019, when the first reports of a respiratory virus outbreak in China occurred, the scientists were ready and willing. So when the sequences were published in January 10th, 2020, they decided on the sequence for the vaccine by January 13th. Moderna started manufacturing the vaccine the following day. The vaccine was tried in mice by February 4th, and the same time that Moderna was making vials for clinical development. Very quickly, the immunogenicity was confirmed in the mice, and the investigational drug application was applied with the FDA. And so the FDA cleared safe to proceed for the phase 1 trial March 2nd. The first phase 1 trial started March 16th of 2020. Just over 2 months later, on May 29th, the phase 2 trial started. And by July 27th, the phase 3 trial was starting, which is all in record speed. And one could ask, were corners cut? And no, the answer is no, corners weren't cut, but we did some very novel things for these trials. And I'm going to talk about how that happened coming up. So the usual vaccine clinical testing process starts in the preclinical phases, where we are testing vaccines in cell culture and assays, laboratory assays, and then going into animal models. We generally start with rodents, mice, and other animals like that, before going into slightly bigger animals, maybe rabbits or other animals, and then eventually going into non-human primates before going into people. For these vaccines, we compressed this a little bit. We did have the animal data for the mice before we went into people, but we continued to do some of the animal studies alongside some of the human studies. So this slide shows the usual clinical trial stages for pre-pandemic life. So before a vaccine goes into people, there's all the preclinical data that's done ahead of time. And then the company would apply to the FDA or another regulatory body for an investigational new drug application. And only when the FDA says it's okay to proceed does it go into phase 1 clinical trials. And the phase 1 trials are often the first in human trials. They are primarily done for safety, although they can be a first look at immunogenicity as well. There are usually somewhere in the tens of volunteers in a phase 1 trial. It depends on how many arms the trial has, but it could be anywhere from 20 to 100 people. And usually it takes time to do the trial, recruit the volunteers, gather the data, follow them for the whole duration of the trial, analyze the data, publish it, and then go look for money for the next trial. So it's usually 1 to 2 years. If the phase 1 trial shows the vaccine appears to be safe in these small numbers, then we move on to a phase 2 trial, where again, safety is the primary outcome, but we also might be looking at different doses, can be looking at immunogenicity and early efficacy. Usually for phase 2, the numbers are slightly more. It could be hundreds of volunteers. And again, because the process just takes time, it usually takes 2 to 5 years to go through the phase 2 trial. And it's not usually until after all of this data is gathered and analyzed and gone over very carefully that we even start planning a phase 3 trial, because phase 3 trials are incredibly expensive. The goal of a phase 3 trial is to look at the efficacy of the vaccine in addition to safety and immune responses. Generally, there are thousands, tens of thousands of volunteers in a phase 3 trial, and the time is usually over 5 years to complete the trial from the time you enroll people and follow them and analyze the data. So this is a very long process. Once the phase 3 trial data is obtained, and if it is favorable, then a company can apply for licensure to the FDA. That application is also an incredibly involved application, requiring thousands and thousands of pages of information on the manufacturing, the stability, the chemical formulations, and as well as the preclinical data and all the clinical data as well. If the vaccine is given licensure, it then goes into use, and even after the use of vaccine, we often will require phase 4 long-term observational studies in the general population. So you can have very, very large numbers of people in these trials. And this is to capture any rare adverse events that you may not see from the original clinical trials. So that was how we developed vaccines normally. And the normal timeline for vaccine development, which is up here on the top of this chart on the left, is somewhere between 10 to 30 years. Prior to the pandemic, the fastest vaccine that we had developed was the mumps component of the MMR, and that was 4 years. And our original goal with the COVID vaccines were to speed it up to 12 to 18 months. And the way we did this was not by cutting corners or not by skipping phases, but we overlapped things. We overlapped the phases. We actually conducted some of the non-human primate work and other animal work at the same time that some of the human trials were being done. And more importantly, we invested a tremendous amount of resources into these trials. So we invested human resources. Everybody who is doing vaccine clinical trials stopped what they were doing, and everybody worked on COVID vaccines. So you had the whole research community all working on essentially the same few vaccines to develop, to recruit and enroll these studies as quickly as possible. At lightning fast speeds. We also, invested a tremendous amount of money and risked a lot of money into the development of these trials. The government and the companies basically said, we will pay what it costs to do all 3 phases, even if it's not successful in the earlier phases, just so that we can get the infrastructure in place. We are planning the phase 3 study while we're just starting the phase 1 study, just so that there is no delay. The other thing that we invested in was developing the manufacturing capacity for the vaccines and the commercialization and scale up of these vaccines very quickly while we were doing the trials, before there was even a proof of concept that the vaccines would work. And the government de-risked this for the companies by promising to buy doses regardless if the vaccine was licensed or not, so that the companies could feel comfortable taking that risk and producing these vaccines as quickly as possible. And it was, like I said, it was incredibly effective. And because we overlapped these phases, because we built the factories early and produced the vaccines quickly, we got vaccines out in record time. So there was over 240 vaccine candidates that had some background work done on previous trials. 54 candidates went into phase 1 clinical trials, 41 went into phase 2, 32 went into phase 3, and there were 3 that were granted emergency use authorization in the United States. And I'll talk about each of these individually. So this is an overview of the COVID-19 vaccine candidates that were into large-scale phase 3 trials in the US. You've heard of the Moderna vaccine, the Pfizer BioNTech vaccine, and the Johnson & Johnson vaccine. They all use a little bit different technology. The Oxford AstraZeneca vaccine uses similar adenovirus vector technology as the Johnson & Johnson vaccine. The Novavax vaccine is a new vaccine that has not yet received emergency use authorization, that uses a recombinant protein and an adjuvant, and the Sanofi product, also in phase 3 trials, also uses a recombinant protein and an adjuvant. And so these are different vaccine constructs that will hopefully also be available at some point. The most important thing is all of these vaccines target the spike protein of the coronavirus, of the SARS-CoV-2 virus. This is a picture of the spike protein. And it is a fusion protein that's involved in the fusion of the cell of the virus to the cells in terms of invading the cells. And it changes its conformation depending on where it is in the fusion cycle. And so what we know is that the most potent neutralizing antibodies, the antibodies that work the best to block the virus from invading, are the ones that target the pre-fusion state of the spike protein. And because of this, most of the vaccine candidates have used recombinant DNA technology to introduce mutations into the spike protein that allow it to stay in this pre-fusion form and not go into the fusion form or post-fusion form, which is less immunogenic and the immune system doesn't see. And so the vaccines that are currently authorized in the US, the Pfizer, Moderna, and Johnson & Johnson vaccines all use a stabilized spike protein pre-fusion. As I mentioned before, there are 3 vaccines that are available in the US. The Pfizer mRNA vaccine is licensed for people 16 years and older, and it is available in the US under emergency use authorization for children ages 5 to 15. The Moderna vaccine is also an mRNA vaccine. It's available for under emergency use authorization for those 18 and older, and an application has been filed for those 12 to 17, but that has not yet been authorized. And Johnson & Johnson, the adenovirus vectored vaccine, is available under emergency use authorization for those 18 and older. So I'm going to take a step back and just talk about how each of these 2 different vaccine technologies work before I describe some of the studies that were done. So the first vaccine that I'm going to talk about are the mRNA vaccines, which is the technology used by both Pfizer and Moderna. And it takes the RNA genetic material of the virus that codes for the spike protein that is stabilized in this pre-fusion spike protein, and they take this RNA sequence for that spike protein and put it into lipid nanoparticles or lipid spheres. The lipid serves as two different functions in the vaccine. They serve as both a stabilizing force for the mRNA to keep it stable until it enters the cells. And it also allows the RNA to enter the cells by binding to the cell membranes of the cells once the vaccine is injected into the arm. The RNA enters the cytoplasm of the cells, where it is met by the ribosomes, which translate the RNA into protein. The protein that is created are the spike proteins, that is found on the surface of the coronavirus. The spike proteins go to the lymph nodes, where they induce a strong immune response that includes antibodies, but it also includes T cells and memory cells that provide really great protection against the coronavirus. The other vaccine that's available in the US is the Johnson & Johnson adenovirus vectored vaccine. There's another adenovirus vectored vaccine, the Oxford AstraZeneca vaccine, that's available in many parts of the world. This works similar to the mRNA vaccines in that the goal is to get the genetic material that codes for the spike into the cells. But these vaccines use a different technique to get it in there. They put DNA, the DNA that codes for the spike protein, into an adenovirus. And the adenovirus is a cold virus that is changed, altered so that it can't cause a cold. It's attenuated, it is non-replicating, it can't copy itself inside of us, and basically serves as just a vehicle to get the genetic material into the cells. When the adenovirus enters our cells, it releases the DNA, which is transcribed into RNA and then translated into protein. Again, the protein is the protein that codes. So it's just, the sequence is the code for the spike protein. And so for both of these vaccines, again, the cells are making spike protein, which then induces a very strong immune response that consists of both antibodies as well as T cells and memory cells. And so again, a really strong, durable immune response to this vaccine. I am going to switch gears now and talk a bit about the vaccine phases and the studies that were done to prove that these vaccines were safe and immunogenic and were effective against COVID. And because there are 3 vaccines authorized in the United States right now, I'm just going to use each one as an example for a different phase, a study of a different phase, and show you some of the data from the studies that were done. So the first one, the first phase is the phase 1 trial, and the example that I'm using for this is the Moderna vaccine. This is the mRNA 1273 trial. They tried 3 different doses of this vaccine: 25 microgram dose, 100 microgram dose, and a 250 microgram dose. This study was initially done as an open label study where everybody got the vaccine. So, and there was 15 people in each dose group. They did 1 dose at a time to see how well it was tolerated. And you can see there was not a lot of diversity in the phase 1 trial, although there was more diversity in the phase 2/3 trial. And because this was a first in human vaccine trial using this vaccine, they used young adults who were generally healthy, just to make sure that the vaccine was well tolerated in this population and induced a good immune response before going on to a larger study. And this is a safety reported from this trial. So all the symptoms from dose 1 on the left-hand side and dose 2 on the right-hand side, with the different dose groups, 25, 100, and 250. And what you can easily see is that there is that the 100 and 250 have more symptoms, with the 250 microgram dose having more severe symptoms, especially after the second dose. And particularly, there's more severe fatigue, there's more fever in the 250 microgram dose, and more chills, with more severe chills in this dose. And because of that, and because of the immune responses, so these are the immune responses to different viruses in either the immune responses by different assays, excuse me, to and the different doses, 25 micrograms, 100 micrograms, and 250 micrograms. And what you can see is that the immune responses after vaccination, 2 doses of vaccination, was pretty equivalent in the 100 microgram group as well as the 250 microgram group. And that led the safety committee as well as the manufacturers and the regulatory board to choose 100 micrograms as a dose for the Moderna vaccine to go into larger phase trials. The 250 increased the reactogenicity or the side effects from the vaccine without increasing the immune responses. And so they felt like this was a sufficient immune response with a 100 microgram dose. And that has held up even in older adults and as well. So that's seems to be the appropriate dose for this vaccine. Moving to the phase 2/3 trial, the example I'm using is the Johnson & Johnson trial. This study was particularly large. There was almost 44,000 people included in the trial in multiple countries. They ended up analyzing not quite 40,000 who were seronegative at baseline, meaning that they had not been exposed to COVID or infected with COVID prior. 19,000 who received the vaccine, and 19,600 who received the vaccine, and 19,600 who received placebo. Again, and they had they combined the phase 2 and 3 trial just for ease, but there was a immunogenicity group in the phase 2 trial as well as a safety subset that looked at the safety reactions much more carefully. This is the result of the results from the safety subset, and the placebo versus the vaccine group versus the placebo. The local events and the systemic events. The Johnson & Johnson vaccine is a 1-dose vaccine, so this is just after 1 dose. And in general, while the vaccine did cause some more side effects than the placebo, it generally was well tolerated with very few severe reactions. The fever rate was relatively low in the population in those who were vaccinated, and not much more than the placebo recipients. And then this is the important graph when looking at how well the vaccine protected against COVID. So in the red lines are the percent, the incidence of COVID cases in the vaccine group, and in the blue line is the incidence of COVID cases in the placebo group. These are the top graph is moderate to severe cases of COVID, and the bottom graph is severe to critical cases. And you can see the vaccine protected really nicely against moderate to severe illness, and even better against severe disease with SARS-CoV-2 after 1 dose. And so based on this data, the safety, immunogenicity, and efficacy data, the FDA granted emergency use authorization for the Johnson & Johnson vaccine. And then the last example I'm going to use is the Pfizer vaccine trial in children age 5 to 11 as an example of the pediatric vaccine trials. So this trial went back and did a phase 1 study in these kids for this age group, looking at different doses of of the vaccine. So adults got 30 micrograms of the vaccine. We enrolled a total of 48 children in this study in the phase 1 to receive either 2 doses of 10 micrograms of vaccine, 20 micrograms of vaccine, or 30 micrograms. Because of the increased side effects that were seen after the second dose in 4 of the volunteers at the 30 micrograms, they did not get a second dose at that high. And we looked at both the safety in these 3 groups as well as the immune responses, and chose the 10 microgram dose as the best dose that provided the best safety profile, but also really good immune responses. And so then we moved on to the phase 2 and 3 trial, where we had enrolled over almost 2,300 kids that were randomized 2-to-1 to receive vaccine or placebo. And over 1,500 volunteers completed who received the vaccine, and 750 volunteers who received the placebo were compared. These are the side effects. Okay, so you can see pain at the injection site was the most common side effect after vaccination for both dose 1 and dose 2. The systemic side effects, such as fever and fatigue, were less than what was seen in the adults at the full dose with the 10 microgram pediatric dose. And most importantly, there were very few severe adverse reactions after vaccine. And the vaccine in the 5 to 11s, the 10 micrograms of 5 to 11, was as immunogenic as the 30 micrograms in the comparator group, which is 16 to 25-year-olds. So it's the same neutralizing antibody titers in those groups in the subset that we were looking of the immune responses in. And the vaccine was very effective at preventing COVID. So these are days since dose 1 on the y-axis. The red is the participants who received the vaccine. The blue are participants who received COVID. So over the follow-up, the brief follow-up, there were 3 cases of COVID in the vaccine recipients and 16 cases in the placebo recipients. Thankfully, none of these cases were severe. None of these cases were hospitalized. In addition to the 3 vaccines that are licensed, that are authorized in the United States, and the few others that are in phase 3 advanced clinical trials, there is many other vaccines that are under consideration worldwide. Worldwide, there's almost 200 vaccines that are still in preclinical development that have not gone into people, 132 vaccines in clinical development, the greatest proportion of which are either protein subunit vaccines, which is a more traditional vaccine manufacturing format, RNA vaccines, non-replicating viral vector vaccines, such as the Johnson & Johnson vaccine, and some other vaccines that we don't have in the United States, such as the inactivated virus vaccine and DNA vaccines. Of those, 32 vaccines have entered large-scale efficacy trials. 15 have been authorized in various countries throughout the world, with 8 that have received full approval in at least 1 country. Not all vaccines are successful, as you may know, and so 10 of these vaccines have been abandoned after clinical trials. Want to spend a little bit of time talking about the difference between emergency use authorization and licensure. So emergency use authorization is not licensure and is a mechanism to facilitate the availability and use of medical countermeasures, including vaccines, during public health emergencies. The FDA may allow the use of unapproved medical products where there are no adequate approved alternatives. And that's really important, because if there were other alternatives, the FDA would not provide emergency use authorization. For the COVID-19 vaccines, the FDA came out with guidelines of what they would consider as the minimum requirements for COVID-19 vaccines to receive emergency use authorization, which is manufacturing information to ensure quality and consistency. The known and potential benefits outweigh the known and potential risks of the vaccine. The primary outcome of the study, usually it's an efficacy study, but in the children, it was an immunobridging study where they were looking at the immune responses in the kids, has been achieved. There has to be at least 2 months of follow-up after the last dose, which is different from the licensing requirements, because for licensure, usually they require at least 6 months of follow-up after the last dose, and sometimes even longer. There has to be at least 3,000 vaccine recipients in a safety database who are capturing safety data very carefully, and often there's many more. And an emergency use authorization is granted after the public meeting of the Vaccines and Related Biological Products Advisory Committee of the FDA, which is a group of external experts that determine whether they recommend that or not. In the end, it's the decision of the FDA commissioner to grant the emergency use authorization or not. Licensure, as I mentioned, requires at least 6 months of data, sometimes longer, and tremendously more details in terms of their application, but it is not a lower quality. Emergency use authorization isn't a lower quality, it's just a faster way to get the vaccine available in the event of a public health emergency. Just want to talk about a few other terms, breakthrough cases and why we care about them. Breakthrough cases are defined as cases of COVID-19 infections in fully vaccinated individuals, and we expect these to happen. We know that the vaccine is not going to be 100% against infection. Very few vaccines ever are. And the symptoms of individuals with breakthrough infections appear to be mild to moderate, less severe, and shorter in duration. They generally aren't hospitalized, generally don't die from it. However, people who do get breakthrough cases can be contagious. So it's important to continue to isolate around them. But most importantly, the majority of individuals who are hospitalized with COVID-19 are unvaccinated. They are not people who've gotten vaccine and had a breakthrough case. And this is just shown in this data from the CDC, where unvaccinated people are more likely to be infected, to be hospitalized, or to die. So this top graph shows the rate of cases by vaccination status. The black is unvaccinated individuals from April through September of 2021. And the blue is the vaccinated people. So this is the Delta surge here that started in July. You can see it peaked really high in the unvaccinated. And while there was a slight increase in the number of cases in those who are fully vaccinated, it nowhere reached the rate of the unvaccinated. And we know that if you're unvaccinated, you have a 6 times higher risk of testing positive for COVID than somebody who is vaccinated. These are the COVID deaths by vaccination status. So again, the black is those who are unvaccinated, and the blue is those who are vaccinated over the same range of months. There is significantly higher risk of dying from COVID, almost 12 times, depending on the series you are seeing. Here it's 11.3 times the greater risk of dying from COVID if you are unvaccinated compared to a vaccinated individual. And then the bottom graph is looking at hospitalizations by vaccine state for 2021. And here is the surge that we had in the spring. And then here is the Delta surge that started in July. Many, many hospitalizations in those who are unvaccinated, very few hospitalizations in those who are vaccinated. We also know that the risk of infection is greater than the risk of hospitalization or severe disease. And we also know that this risk is greater with the Delta variant. So these are studies. The blue is the risk of breakthrough is the vaccine efficacy against any sort of infection. And the red is vaccine efficacy against hospitalization. And even though there's lower efficacy against infection after Delta, the rates of hospitalization in those who are vaccinated is really good. And even in people with underlying medical conditions, even with the Delta variant, they still have 80 to 85% vaccine efficacy if they responded to the vaccine. If you're completely immunosuppressed, it's harder to respond to the vaccine. This is another graph looking at the efficacy of the vaccines against infection, and you can see that the efficacy against infection is really, really good until Delta hits, and then the vaccine efficacy decreases somewhat. But the vaccine effectiveness against hospitalizations remains very, very high, no matter what your age group. Even in people 75 years of age and older still have over 90% vaccine efficacy. And there's no significant differences in vaccine efficacy by age group or calendar month of hospitalization. We know that those who are immunosuppressed are less likely to respond to vaccines. This is a small study looking at antibody responses in people with pre-existing conditions. So if you have cancer, especially hematological cancer, you're not going to respond as well as somebody who has a normal immune system, where the antibody responses are somewhere in the order of 95 to 100% of individuals will respond with good antibody titers after vaccination. Dialysis patients, there's some variability in their antibody responses, but some have significantly lower antibody responses, as do people who are on immunosuppressive therapies, such as people who have rheumatological or autoimmune diseases. But the population that has the lowest antibody responses and the longest difficulty are the people who've had a solid organ transplant, where the majority of them don't respond after 2 doses of vaccine, and only about half will respond after a 3rd dose. And it's really based on data like this that the CDC has recommended a 3rd dose of vaccines in people who are immunocompromised, moderately or significantly immunocompromised, people who have solid organ transplants, people who are on medications that suppress their immune system tremendously. And this is not a booster dose. This is a 3rd primary dose of vaccine to be given about 4 weeks after the first 2 doses to raise the antibody levels. These people are still potentially eligible for a booster dose as well, which is going into the next slide, where the FDA and the CDC recommends that people who receive the Pfizer and the Moderna vaccine more than 6 months or more than 6 months prior should consider getting a booster if they are over the age of 65 or they are 50 to 64 with underlying conditions that put them at severe risk for COVID if they get it. Other adults who can get the booster include those with underlying medical conditions who are younger, or those who are at high risk exposure due to their job. And at the time that I am recording this CDC ACIP will meet in just a couple of days, and as will the FDA, and likely recommend boosters for all adults 18 and older. The recommendation is that those who have received the Johnson & Johnson vaccine will get a second booster as well, and anybody who's received the Johnson & Johnson vaccine can get a booster. You can mix and match the different vaccines and get 2 different varieties if the government will. You can mix and match the vaccines, and that there seems to be some improvement in the immune responses if you're using a different vaccine to boost compared to the first series. And overall, boosters seem to be well tolerated with symptoms similar to that seen after the 2nd dose. The reason we need boosters is that as we age, our immune system has less ability to respond to new stimuli. When we are infants, we are born with a fully functioning thymus. We have a lot of naive T cells and very few memory cells. But over the course of our lives, as we are exposed to more and more foreign antigens, we're creating more and more memory cells. So by the time we get to our older age, and our thymus involutes and becomes dysfunctional. So by the time we get to older age, we have lots and lots of memory cells, but there's fewer naive T cells that are available to respond to new antigens and to respond to new stimuli. So as we get older, our ability to form new memory T cells is impaired. And so therefore the durability of the immune response in older adults is likely not as good as the durability of response in younger people with a healthier immune system. I just want to add that we have had a significant drop in life expectancy in the United States, especially compared to peer countries, because of the COVID pandemic, and the hardest hit communities are communities of color, where there is a lot of vaccine hesitancy. And so it's really important to talk about the risks of COVID in these populations, as well as address any concerns that they may have about the vaccine, so that people are willing to accept the vaccine and to take it. So you can see here in our peer countries, there really hasn't been much of a decrease in life expectancy, even with the COVID pandemic. But for the US population, the US total is in the yellow line here. For US Hispanics, the life expectancy has dropped to almost 4 years because of the pandemic. For white, non-Hispanic whites, it's a little over a year, and non-Hispanic Blacks, the life expectancy has decreased by almost 3 years. And this is really quite shocking, and really even more reason to continue to encourage people to get the vaccine. And then finally, I want to finish up by talking about the US vaccine safety monitoring system, and how really impressive and really thorough the vaccine safety monitoring system happens in the US. So there's multiple different modalities to monitor vaccine safety. The VAERS is a vaccine adverse event reporting system. It's a passive reporting system. That means anybody can report to it for any reason. You can report that the vaccine made your toe turn green and start sprouting flowers, and VAERS would accept it because there's no filter for causality or other things. And there can be duplicate reports in VAERS. There can be mistakes in the reports in VAERS. So it's a passive system that anybody can report to, that just allows for signal detection, but really to look at different adverse events that are particularly of interest, and that we can start looking at association and potentially causality. There are multiple systems, including the Vaccine Safety Data Link, that uses large healthcare networks to examine claims data. And the Clinical Immunization Safety Assessment Project is a project of the CDC and 7 academic medical centers to provide clinical safety guidance to US physicians or practitioners anywhere. So real-time questions they may have about their patients and whether to give them vaccine or what vaccine to give them. CISA is available to answer these questions. In addition, there's been a new vaccine safety monitoring system. It's been put in place just for the COVID vaccines. It's called vSafe, which is a new smartphone-based app, a monitoring system for individuals who get the COVID vaccine to record adverse events after vaccination. If you register for vSafe after you get your vaccine, you'll get a text daily for the first 7 days after each dose, to remind you to answer questions about vaccine safety, and then weekly for 6 weeks. And if you happen to be pregnant with the vaccination, they will enroll you into a registry to follow the safety of the pregnancy. And I'm going to end here, and thank you for your attention, and please contact me if you have any questions.